Okay, so we have talked about resting membrane potential. Resting membrane potential is primarily because of leak channels. And now we've talked about graded potentials, and graded potentials are because of gated channels. But I want to think about ligand gated channels that we saw in the last video so that you can have a visual of how to open those, because I want to distinguish it from what we're going to talk about next, which is voltage gated channels. When you get a big enough depolarization here to cause the axon hillock, we're talking about neurons here, to hit threshold, which averages about minus 55, and then you open a new kind of channel, and the new kind of channel is a voltage gated channel. Okay, so when you open a voltage gated channel, you're at a new kind of potential called an action potential. Okay, and an action potential is basically this self-propagating potential that will go all the way down the axon. It's going to spread down the entire cell membrane of a neuron, and it goes in other cells too, but we're going to focus on neurons first. So the process basically goes like this. So let's look at this one. Okay, so if you start with a neuron, poorly drawn, like I draw everything, then the whole thing, the whole blasted neuron, of course, would have been at resting membrane potential, right? So that's minus 70, okay? And then let's say, just to sort of correlate with the picture that we were looking at before, let's say that we opened ligand-gated sodium channels here. We're going to open them here, okay? And the positives are going to go inside, inside, inside. So sodium's going to go inside. And that would, of course, be depolarizing. So what you would be seeing in the graph right now would be like you started at minus 70, you start going up. Okay, so what happens, the magic will happen, is if you get this area over here, which is called the axon hillock. If you get that to minus 55, which is threshold, then you'll get an action potential. Okay, so you get an action potential there. So what you need to do is you need to have a big change over here to cause this change over here. So if you hit threshold at the axon hillock, then what's going to happen is you're going to open a new kind of channel. And the new kind of channel will be a voltage-gated sodium channel, which are really clustered at the axon hillock. Okay, so if you open the first voltage-gated sodium channel, you're golden, because I want you to think of them as lined up along the axon hillock like dominoes. Like here's one, here's one, here's one, here's one. So they're lined up like dominoes, and they're all voltage-gated sodium channels, and they'll all open in response to being depolarized to minus 55. They will open. But here's the important thing, and here's why it counts as a positive feedback example. Because if I depolarize this voltage-gated sodium channel to minus 55, then it will let enough sodium in to make sure that this one gets to minus 55. And that will let more sodium in to make sure that this one gets to minus 55, all the way down the axon hillock. So if I open the first one, if I depolarize the one that's right here, it will make sure that the next one opens, and the next one opens, and the next one opens. So basically what we have is sodium rushing in causes the depolarization that causes sodium rushing in, that causes to depolarization, that causes sodium rushing in, that causes the depolarization. And each channel opening adds to the depolarization and makes sure that it continues to go down the axon by opening more and more and more of the dominoes all the way to the end. The change is really, really dramatic. It looks like this. So from here to here is a graded potential. That's all what we talked about before. But the magic happens if you hit minus 55, because then you open the first voltage gated channel. When you open the first voltage gated channel, you're going to get a massive action potential. So the change is really dramatic. You started at minus 70 and you can go all the way past 0, all the way up to positive 30. Okay, so now I've got this massive change. So let's take a second and make sure we know what's actually going on here and just do a little labeling, and then we'll do some complete labeling in just a second, but I just don't want you to get lost. Okay, so let's talk about what is actually happening here. So same questions I've asked you before. What kind of channels are involved in getting me to minus 70? What kind of channels? Those are leak channels, right? Exist in every single cell. What kind of channels are involved in getting me from minus 70 to minus 55? Those are, okay, they have to be sodium because it's depolarizing. And they have to be gated because it's not staying at resting membrane potential. And I want to call them ligand gated because I want you to imagine that it's like acetylcholine binding to and opening those. Okay, but now question is, what kind of channels are involved in getting me from minus 55 all the way up to positive 30? Those are voltage gated sodium channels. Are they opening or closing during that? They are opening. Okay, so all right, so now let's talk about the repolarization that happens after the action potential. So how do I get from here back down to minus 70? How do I get there? Well, hopefully you will know that in order to do that, what I'm going to need to do is at the top here, to start moving down, I'm going to have to do two things. I should close my sodium channels, voltage gated sodium channels. And then what's a really quick way to make me go all the way back toward minus 70 from 0 or positive 30? What's a quick way to do it? Remember that repolarization was generally caused by opening potassium channels, because what I just did is let a whole bunch of positives in. I'm going to close that, and now I'm going to let a whole bunch of positives out. So to go all the way back down here, actually it goes all the way down to there, this is open, and they are voltage gated too, voltage gated potassium channels. That will allow for repolarization. And that's all well and good, with the exception of the fact that everything's fine, and I may have gotten back to minus 70, but all of my ions are in the wrong place. I just reestablished the positive negative nature. It actually kind of overshoots it just a little bit, but then it like settles back at minus 70. So this was all sodium moving in, this is all potassium moving out. Then I'll settle back at minus 70, but it's not homeostatic to have that much sodium in, and it's not homeostatic to have that much potassium out. So I fixed the number back to minus 70, but the ions are all in the wrong place. So what do I have to do now to fix the location of the ions? What do you think? Yeah, it's our friend, the sodium-potassium pump. Now it just has to change the position of the ions. It really doesn't change the number dramatically when it does that. So a bit massively depolarized here with letting a whole bunch of sodium in. I massively repolarized with having a whole bunch of potassium out. And then I'm just going to pump my little axon buns off to get everything back into position. Okay, so last but not least, we're going to draw this, and then I'll show you a short video that gives you a visual of all of this. And then we'll be done with action potentials. This stuff is difficult though, so you're going to probably need to review it. So let us do this on your notes. So let's read. On the diagram below, sketch the membrane potential recorded in a neuron as it depolarizes to threshold, undergoes an action potential, and then repolarizes to resting membrane potential. So lots of information in there. And of course, I'm going to ask you what kind of channels are involved in every single place because I'm annoying like that. So let's do this together. All right, here we go. Okay. I am going to listen to the instructions and I'm going to say, okay, sketch the membrane potential recorded in a neuron as it depolarizes to threshold. So it's definitely going to start at resting, right? And it's going to depolarize to threshold. What is threshold? Threshold is right here, so I have to go that direction, which is depolarizing. Undergoes an action potential. Action potential goes all the way up to positive 30. And then repolarizes back to resting membrane potential. It always slightly overshoots and then comes back up to resting membrane potential. That's supposed to be at minus 70. Okay, so I've done it, but now, of course, I want you to label the whole blasted thing. So I'm going to ask you the same questions that I asked before, which is, okay, what kind of channels got me here? What kind of channels get me to resting membrane potential? Hopefully you know by now. That was leak channels. Next question. How did I get from resting membrane potential to minus 55? What kind of channels are involved there from minus 70 to minus 55? What are those? Those are opening. What kind of channels? Well, they're definitely sodium, but let's add a little bit more detail. Let's call them ligand-gated sodium channels. And then if I cause a massive enough voltage change to hit threshold, then what kind of channels are going to be involved here? These are opening. This time they're voltage-gated sodium channels. Okay, but now I'm at the top. So now what do I do right here at the top? I've got to do two things. I got to close something to stop what I was doing, and then I have to open something else. So what happens here? I'm going to close voltage-gated sodium, and then in order to get all the way back down, I'm going to open voltage-gated, oops, not that, silly Melanie, potassium channels. Okay, and not shown on the diagram because it really doesn't change the number, I have to also run the sodium-potassium pump. It's happening before, it's happening during, it's happening always. Okay, so now let me show you an animation that slowly and surely, and if you don't like her voice, I always hear that. I'm sorry. But it's a good visual. It's a nice little animation for you. And then we'll be done with this.
Neurons send signals over long distances by generating and propagating action potentials. Most action potentials originate near the axon hillock of the cell body in the initial segment of the axon. It then travels the entire length of the axon. A closer look reveals that during an action potential, voltage-gated channels open and close, altering the permeability of the plasma membrane to sodium and potassium ions. A threshold stimulus changes the shape of the voltage-gated sodium channels, causing their activation gates to open. This event marks the beginning of phase 1 of the action potential, known as depolarization. As sodium ions diffuse into the axon, the membrane potential becomes less negative. This causes more voltage-gated sodium channels to open, and the membrane potential soars to plus 30 millivolts. At this point, two key events occur. The inactivation gates of voltage-gated sodium channels close, and voltage-gated potassium channels open. These two events mark the beginning of phase 2 of the action potential, known as repolarization. As potassium ions diffuse out of the axon, the membrane potential becomes negative again. However, the membrane potential continues in the negative direction, going beyond the resting state of minus 70 millivolts. This marks the beginning of phase 3 of the action potential, known as hyperpolarization. During this phase, voltage-gated potassium channels close and all voltage-gated sodium channels are released from inactivation. By the end of this phase, ions move through leak channels only and the membrane potential returns to the resting state of minus 70 millivolts. The neuron is now ready to fire another action potential. Summary: generation of an action potential. A threshold stimulus opens voltage-gated sodium channels. Sodium ions diffuse into the axon, depolarizing it to plus 30 millivolts. Voltage-gated sodium channels close, and voltage-gated potassium channels open. Potassium ions diffuse out of the axon, repolarizing it to a negative value. The membrane potential briefly hyperpolarizes. Voltage-gated potassium channels close, and the membrane returns to the resting state of minus 70 millivolts.
Okay, done. Next, we will be talking about how to get the second action potential after the first one.